My name is Lauren. I'm a PhD student at UCLA, and today I'm going to talk to you about competitive interactions between Sargassum Hutterites and invasive algae here, and native algae under varying temperature regimes. Now, before I get too deep into my love for competitive interactions between algal species, I want to talk to you a little bit about how I got here. So I grew up in Dana Point, so not too far away. I rock the 90s beach look. I'm the oldest of 3 kids, and my family spent a lot of time at the beach, and I quickly developed a passion for the ocean and all of the creatures within. I spent my summer at 5 throwing sea stars and sand dollars back into the ocean on the beach in South Carolina. At 10, I was attending ocean camp at the Ocean Institute and collecting as many crabs as I could in my baseball cap. And by high school, I was pretty sure that this marine ecology thing was going to pan out for me, and it was going to be what I wanted to do long term. So I actually was really lucky. My high school has a marine ecology class, and so in high school I took marine ecology, and after spending 9 months, is that how long you're in school? Learning hundreds of genus and species names, how to identify algae from really far away, and all the seashells in the area, I got to go on a 9-day field study to Bahía de las Animas, Mexico. And so while we were there, we worked in groups, and we got to explore all these different habitat types. So we have mangrove forests and mud flats and a soft coral reef, sandy reef, and a mangrove forest. And so there was my real first experience getting out into the field and really just exploring what it meant to be a marine ecologist, and I thought to myself, this is something I could get behind. And so just a couple of pictures here. This is a jellyfish. This is a sunburst sea star. I think this is a chocolate chip star, and these are blue swimmer crabs. They're pretty aggressive. So if you see them, don't challenge them to a duel. Once I graduated from high school, I went to college. So I went to UC Santa Cruz, and if you've never been, it's a great location to do marine science. They have the Long Marine Lab, which I pictured here, and a bunch of just really super cool classes. So I took classes with titles like marine mammals, coastal conservation ecology, marine botany, kelp forest ecology. And while I was there, I actually got to dive. I became a scientific scuba diver. And so I took a class where I got to design my own experiment with scuba, and I got to go out into the Monterey Bay and conduct an experiment with scuba, and I thought, this is awesome, and why would I ever not do this? And so it was a really cool time, and I spent a lot of time here at the Long Marine. So Santa Cruz has a main campus that's up in the forest, which is amazing, but then they also have this Long Marine Lab, which is just right down on the waterfront, and just, I don't know, just a lovely place to be. And so I got really excited about, especially California coast. And while I was an undergrad, I was given a wonderful opportunity to study abroad. So I traveled to University of Queensland, Australia, where I got to hang out with kangaroos, go to the Sydney Opera House, but also do 2 more field studies on the Great Barrier Reef and North Stradbroke Island. So while I was at the Great Barrier Reef, I watched crabs eat algae off of coral, which is riveting. But we were trying to show that there's an interaction between algae and coral, where algae can grow much faster than coral. Coral is a very species, and so what was keeping that algae from outcompeting that coral and growing up over it? And so we set up all these little jars, which I'm going to get back to jars and mesocosms in my talk. But we had little pieces of coral and algae in each one, and the crabs in there, and they just slowly ate off of the coral. It was a lot of fun, and that's kind of when I knew that graduate school was probably on the table for me. So when I got back to UC Santa Cruz, I started looking for a lab to join, and I found one in the Coastal Conservation Action Lab. So I started working with a graduate student named Dina Spatz, who was building a database. So this is a picture of the database website. It's the Threatened Island Biodiversity Database, or TIB. And you can actually visit this website. It's live online. And what we were doing was prioritizing islands for invasive species eradication. So the way we did that, we worked working with the IUCN, which makes the red list, which is the list of critically endangered and endangered, and island conservation. And the IUCN provided us with this list. So we had a list of critically endangered and endangered island breeding vertebrates. So birds, amphibians, reptiles, and mammals. And with that list, we went through and just poured through the research to figure out what islands these animals were breeding on. And so for things like mammals, it's really easy. If the animal lives on the island, it breeds there. For birds, it's really hard. And so we spent 2 years building this database, and so now you can actually go online. You can pick a region, you can pick a species, you can pick an invasive, and it'll give you all this information per island. So once we had this list of islands that had these endangered species on it, we figured out what threats they had there. And one of the biggest threats is invasive species. And so, especially in an island community where everything is kind of in a really good balance, you have this problem with invasives. And so one of the reasons they target islands is because they have the greatest concentration of both biomass and extinctions. They make up only 5% of the Earth's landmass, but they house 40% of the endangered and critically endangered species. And on top of that, 80% of known extinctions have occurred on islands, which make them really good spots to kind of do this work. And so they pick islands based on how many humans there are, what invasive species were there, to then go in and eradicate an invasive. And they've had huge results that are just astounding. In places where they've eradicated goats, plant species they thought were extinct came back. And so it just is pretty rewarding, and it's the first time that I got introduced to really what an invasive species was. So invasive species have a lot of names. People call them ED, exotic, introduced, non-native, and they have a lot of definitions, but one that's pretty common is that they're not native and they cause harm. So invasives can arrive in a lot of different ways, but a lot of it is due to humans. So we move things around, pets, agriculture, ballast water, just hitchhikers, and things like that. And of the species that have known reasons for extinction, 54 include impacts from invasive species. So they're reshaping ecosystems worldwide. They lead to a loss of biodiversity, ecosystem services and functions, and just cause a lot of problems. It's estimated that in the United States alone, every year we spend $128 billion on invasives. So that's damage that they're causing, eradication, or like efforts to try and abate what's happening. And so they are a real issue, and it was something that I got really kind of excited about and interested in. Once that project finished, I was just graduating, and I wasn't sure exactly what I wanted to do, but another member in that lab was in the Philippines and was looking for divers. And so I thought, well, that could be fun. So I did that. So 2 days after graduation, I got on a plane and I moved to the Philippines for 3 months, and we were doing seagrass research there. So I got to do a lot of fun diving and visit the rice terraces, but I also did a lot of counting grass. So this is a quadrat that we have underwater, and we would count all the blades of grass in a fourth of that quadrat. Then we'd bring it back and we'd measure blades of grass, and we'd weigh blades of grass, and we'd dry blades of grass. But we were looking to figure out if places that had a marine protected area or a terrestrial, like a lack of runoff coming into it, was more beneficial for the health of the seagrass beds. And we found that terrestrial runoff is really what's impacting the health of the seagrass beds. So after 3 months, I returned to the United States. I knew I wanted to go to graduate school. I had a lot of experience with graduate students, and I knew it was something that I was really interested in, but I wasn't really sure what my question was going to be. So I decided to get a job and join the workforce. I actually got 4 jobs, but I'll talk about 2 of them. First, I worked at the Ritz-Carlton as an eco-adventure center naturalist. So we took guests whale watching, snorkeling, tide pooling, and there I really got to work on my educational content and like sharing my passion for the ocean with other people. But that wasn't enough, so I started working for Lagoon Ocean Foundation. So they do tide pool education in Lagoon. So I got to take visitors out into the tide pools and show them what was there and teach everybody how to use your science finger to touch things and just be really kind to the animals. And it was during that job that I started seeing some really big shifts to the places that I loved and started getting concerned and started asking questions. So first, in 2013, the sea stars started getting a wasting disease. So from Alaska down into Mexico, we lost almost all of our sea stars in the tide pools. And this wasting disease coincided with a heat wave. and the sea stars disintegrated. So they'd be there one day, and you'd come back 2 days later, and they'd look kind of like this puddle of sea stars. And just within days, they were gone. And it's pretty heartbreaking to have to tell a family that's traveled 2,000 miles to see a sea star that they're not in the tide pools. Next, we kind of got this pelagic crabs. So they showed up. They're from Mexico, but they come up with warm water. So we just had hundreds of pelagic crabs everywhere on the beach. Did you see them?
Yes, they were last year.
Yeah, they're pretty cool, but different. I'd never seen them before. And then in the tide pools, we started getting sea urchins with the Baldies disease. So you'll notice that it's missing its spines here, and that makes them really vulnerable to predation. So once they don't have spines, seagulls can actually pick them up and drop them, and then they can get at their insides. And so we lost all of the urchins in the tide pools. It was a sad time. And then lastly, I started noticing a new species in tide pools. So this right here is a juvenile Sargassum Hutterites. So I'd never seen it before, and all of a sudden, here it was in my tide pools with all these other things going on, and I started getting really interested, and I started doing some research about it. And so this is a little bit about Sargassum Hutterites, large brown algae. It can grow up to 3 meters in height. It's an annual species, so right now they're all about this big out in the cove, and you get kind of mats of them. So there's a lot at once, and then they go through their growth cycle. They become reproductive. They have both male and female reproductive parts. So they're highly fecund, and they can self-fertilize. Then they go through a senescent period. So they die, and they kind of break apart, and then they start coming back again. And so they're really well adapted for colonization. In 2003, they were found in Long Beach Harbor. Likely they were introduced by a shipping vessel. So at that time, it was localized. It was just in one location. 3 years later, they were here at Catalina Island in 3 locations, likely brought over by pleasure boats from Long Beach Harbor. So today, Sargassum Hutterites can be found 750 kilometers. So from Santa Barbara, Point Conception, all the way to Isla Natividad, and there's at least one report of Sargassum above Point Conception. So that's a little startling, and so I decided this was something that I really wanted to concentrate on for my PhD research. So what am I doing here at the Wrigley Marine Station? I'm trying to determine if temperature impacts the competition between this invasive species and our native understory algae. A couple of years ago, Dr. Lindsay Marks did an experiment here where she removed sargassum from plots, and after her experiment, there was actually more sargassum in the removal and the control plots than there were when she started her experiment. And that was happening during the El Nino, so the water temperature was increased. And so I wanted to look at that question and look at why did this happen. So our native kelp-forming species really like cold, nutrient-rich water. And so I wanted to look at this question and see if I could figure out whether or not sargassum gains a competitive edge once that temperature is raised up a couple of degrees. So the way that I wanted to do that was set up a mesocosm experiment. So I have 30 jars, which are represented by these lovely red and blue circles. And in my jars, I'm pairing algae either with itself, so Sargassum Hutterites with Sargassum Hutterites, or native algae with native algae, and they're all going to weigh the same. Or Sargassum Hutterites with a native algae, and comparing growth in those two over time to see kind of who's growing faster and what some competitive interactions might be. Now, I've shown, I have blue ones and red ones to show that I was also planning on heating up half of those tanks. So I was going to have 5 that of each type that were ambient seawater and 5 that were warm seawater. And so I was going to compare the growth of those two things. But what happens in science sometimes is you're foiled by the weather. So we had a big heat wave. So this is actually a graph of one of my temperature loggers. I put it out. It was about 25 feet down. And this was the course of 5 days. The bottom temperature went up over 4 degrees Celsius, which is almost 8 degrees Fahrenheit. And so my jars were outside, and so they were not only getting this temperature increase, but they were also getting some sunlight. And so the jars were swinging in temperature about 10 degrees every day. And so I took a step back from that and decided that I wanted to do a little bit of field research as well. And so I went out into the field and I started tagging individual sargassum little babies. And so I went down and I put zip ties and flagging tape on them, and I measured how tall they were and how wide they were, and I put temperature loggers down, and every 5 days I went back and I switched out the logger and I remeasured them to see if they were, how they were growing, if the temperature kept changing, how that growth was impacted by that temperature. So here you can see, so this was the time, like the first time step, second time step, third time step. So the temperature actually increased about 2 degrees, 1 degree every 5 days, which is convenient. And then the growth also increased. So I graphed those over each other. And so this is actually temperature here and growth here. But they also correlate. So I got an R-squared of 0.897, which means 89% of the change is due to this interaction, which is pretty exciting. So now I've put out 10 more. I'm going to keep doing it, and then I'm going to do 10 more at another location to see if I can continue getting this line trend. And now it's cooled down a little bit, and I moved my jars inside, and they're much happier. So what I'm doing now, after I have a little bit of progress with my mesocosms, this is what they look like. So I have, these is 12, but I have 15 jars set up like this, and inside they have algae, usually 2 species. This is a temperature logger. This is the heater. These hoses are bringing in fresh seawater, and then I have a min-max thermometer, so I can see how the temperature is fluctuating every day, for the ones that don't have temperature loggers. And so just this week, I finally got these up and running, and I decided to hold off on changing the temperature for right now, since the water's still a bit warmer, and just do a comparison. And so right now, they're full of Sargassum Hutterites and Sargassum Hutterites, which is a native sargassum to this area, and I'm interested to see how those growth changes with those 2 species over time. And then one more part that I'm going to add is going to be in the field. I want to look and make sure that these species are actually competing, right? So if I put them in a jar, I can say, yes, this grows faster than this one, but what if they're never found together in the wild? That won't mean anything. So I'm planning on going out and conducting an experiment over time to see what Sargassum's nearest neighbors are as it grows and changes from being kind of in a mat to being an individual that doesn't have as many Sargassum Hutterites around it, but maybe some other species as well to show what they really do. So I don't have very many results to share with you today, but this is what I'm working on, and I'm pretty excited to see where it goes. So just a couple of thank yous. I really want to thank UC Wrigley for letting me come, and the staff here. They've been really amazing, and the other fellows. Santa Monica Bay Audubon Society, who's helped fund my research. My advisor, Dr. Peggy Fong, my committee members, Dr. Lloyd Smith, Dr. Kraft, and Dr. Kavanaugh, my dive support, Emily Reisner, who's been a superstar, and the rest of my Fong lab. So now I'll take any questions.
Do you have the jars here?
I do have the jars here. And they're upstairs.
Where did this invasive species originate?
It's from Asia. So I think South Korea and Japan.
So when you're talking about your previous research, when you say that you would go in and eradicate the invasive species, what does that mean? Like for goats, do you remove them from the island? What is that?
So for goats in particular, they kill them from helicopters.
Okay.
That one's fun. It depends on what the animal is. So with cats on the Channel Islands, rats they don't remove them. They don't use helicopters. But yeah, that is a Garibaldi.
On our tidal areas, they're gone. Whole Southern California area, you won't see urchins anymore?
No, they've come back.
They've come back.
Yeah, so it took a while, and I'm not sure how Catalina was impacted, since it's a little bit off the coast. But I know in the type pools that I worked in, the last count I got, they had like 32 when they did their surveys. So they've made a recovery.
They've made a recovery, and is there analysis on what happened? I mean, you're trying to tie it possibly to this, or?
No, it's just like the worm part of it was something that made me think about like how sea temperature warming was impacting just kind of the environment in general. So my question is a little bit separate from that, and I believe they think that two things happened, like a virus and a bacterial infection. So once they were suffering from one, it was really easy to come in. and kind of have that wasting happen.
So since your studies following the impact of what seawater temperature has, are you kind of wondering if maybe adding a wish list of where you could further control the temperature of the water more than you do now? Because it sounded like it was a little out of control.
Yeah.
More than you wanted it to be.
Yeah, it is. That would be nice. And sometimes in science you get those things, and sometimes you don't, and you work with what you have.
Everything's great. You know, you always learn something, but I'm just curious if you like, is there maybe more control than you have over the water?
Yeah. I don't have the ability right now, so but it's okay.
When you were showing when like the animals were sick, what were the diseases?
So there's a wasting disease. So it was just called sea star wasting disease. And I'm not positive of the urchin one. They're very similar. They're both echinoderms. So they got a balding disease, but I'm not sure what its name was.
How are they still alive now?
My understanding is, at least in Monterey, that the adults did a massive recruitment. So they had a bunch of babies. And so once the illness had kind of moved on, the babies were able to.
So maybe you covered this, but are you also studying the actual cell biology, the difference?
Mine's just going to be in the environment. There are people who are doing stuff more like that, though.
What's the status now with the increased water temperature, and it seems like it's more long term over the last few years?
Yeah, I don't think anybody knows exactly what's going to happen. I think a lot of people are predicting that the tropicalization in California is starting, and so things that are going to have a warm water affinity are probably going to do better long term if this continues.
And what's the status with the kelp now?
We're seeing some come back here at like Isthmus Point or Reef. They have like a number of juveniles, which has been really fun to see. So they're having some recovery, and I know there are people who are watching that with satellites and stuff.
Thank you so much.